Right, good afternoon, Year 12. So, today we're going to carry on, and this should be quite a nice short lesson on non-disjunction. Now, non-disjunction is the final kind of, you might almost call it a mutation, though it's not a change in the basis, so it's not technically a mutation. But it's the final kind of genetic mistake that can happen that we're going to learn about in Year 12. And very broadly what you're looking at here, you're looking at something going wrong during meiosis so that the homologous chromosomes don't separate properly. And you can see this is happening during meiosis 2. So have a glance at the diagram there on your screen. You see what should happen on the left, possibly your right, is that during the first section of meiosis, meiosis 1, the homologous chromosomes should separate apart, and the maternal should end up in one, the paternal should end up in the other. And during the second stage of meiosis, meiosis 2, those chromatid arms should separate to create 4 cells, each with the same amount of DNA, but each slightly different. In a non-disjunction event, we're looking at something going wrong in meiosis 2. So what happens is that whilst the centromere is pulled apart, both chromatids get pulled to the same side of the cell. And so you end up with one of these cells being created which has twice as much DNA on one chromosome as it should, and one cell being created which has none of one of the chromosomes at all. And this can cause all kinds of issues. So, what I'd like you to do is I'd like you to draw that non-disjunction diagram down, and I've realised that note at the top is slightly misleading. You're looking at the failure of chromatids to separate properly during meiosis, rather than homologous chromosomes, and that's my mistake. So pause me, get the failure of chromatids to separate properly during meiosis down, and then copy down the non-disjunction diagram. Now, you're not required to know anything about what causes this, why this happens. It tends to be an issue either with the centromere itself not separating correctly, i.e. It's taking too much energy, perhaps from too much pull from the spindle fibers, or just a mistake with spindle fibers themselves, the centrioles. So, let's have a look at a couple of questions about this. Firstly, hopefully you should have already written down those notes from earlier, so you don't need to write down the first 2 questions. But have a glance at them. What are the answers? So, a non-disjunction event we hopefully know is a failure of the chromatid arms to separate properly during meiosis 2, which answers question 2 as well. It's likely to occur during meiosis. It's more or less likely to have an effect than gene mutations. Why? Well, the answer is it will have an enormous amount more effects than gene mutations. Whilst none of the DNA you get is mutated or wrong, you suddenly have twice as much DNA on one of the chromosomes as you should. It's the difference between replacing a few letters in a recipe and doing every step twice. If you do every step twice, you cook it for half an hour and then you cook it for another half an hour, you will just not have a cake at the end of your recipe. If you misplace a few letters, it might affect things, but it's a lot less likely than doing every step twice. In the same way, if you have twice as much DNA as you should, you're going to create twice as much protein as you should, twice as much active RNA as you should, and it can cause enormous issues. Now, most of the time non-disjunction events in meiosis lead to a failure of the embryo to develop. But there are there are a few different conditions that occur when this happens and the embryo does develop successfully. I'm going to look at a couple of these. I want to make it clear you don't need to know the details of these at all. So let's look at 1 or 2. First is Down's syndrome. I should say Downs rather than Down. Also known as trisomy 21. Called trisomy 21 because it's caused by the presence of a third copy of chromosome 21. So if we go back to our diagram, what's happened here is this blue one here, down at the bottom, has twice as many chromosome 21s as it should. Remember, this is going to be a sperm or an egg, and so when it gets to meet the other one, either the sperm or the egg, depending on what it is, that will also have a chromosome 21, and you will end up with 3 of these blue chromosomes. And that leads to trisomy 21. So, there we are. Now, we will all have met people with Down syndrome, and so you have kind of an idea of the kind of conditions it causes. It tends to be things like a mild to moderate intellectual disability, learning disabilities, some facial features which are very characteristic, and also physical growth delays, as well as also occasional often problems with the heart and various other organs, which can cause major problems for people with Down syndrome. Now, obviously, this is entirely survivable. There are lots of people living very happy lives with Down syndrome, but it does cause issues. And unfortunately, the major issue for their lifestyle is this intellectual delay. You're looking at most people with Down syndrome having the mental ability of somewhere around an 8 or 9-year-old child. But there is a lot of variation there, and as I say, different people are different, like with everything. The other one we're going to look at is Turner syndrome, which is an example of one of these, which are actually more common, where the sex chromosomes are either not present in the correct numbers, or you have too many of them. Now, in this case, you only have this is a girl who only has one X chromosome. So if I scroll back up to my diagram again, you're now looking at this cell, bottom right here, the empty one. So when we've been doing meiosis, both X chromosomes have ended up over here, and there's no X chromosome in this egg. So when a sperm comes along with its X chromosome and fertilizes it, the zygote will have one X chromosome but not 2, and that leads to Turner syndrome. Now, Turner syndrome causes lots and lots and lots of different things, including a webbed neck, particular shaped face and hairline, swollen hands and feet. But the major issues, people, because all of those are dealable with, the major issues are things that were affected with puberty. Girls who have Turner syndrome tend not to go through puberty normally, and they'll only develop breasts, menstrual cycle, any other characteristic female puberty changes if we give them hormone treatments. And usually they're unable to have children. Now, those are just 2 examples of a lot of different conditions, and particularly with the sex chromosomes, because you can more often survive with either less or more sex chromosomes, there are quite a few of these. So you've got your classic super male syndrome, where you have 2 Ys and an X. You have a lot of possible combinations. But this is all down to non-disjunction events. Now, non-disjunction events, there are none that you need to know specifically, but the one that is going to come up a lot as an example in tests is Down syndrome. So if you already have an idea of what causes Down syndrome, that will probably help you out quite a lot if this does come up. Now, the more common question, however, is this, a data question. Now, data questions tend to stump people in our 6th form. I'm not quite sure why. I think every school has a particular type of question which either the teachers don't teach very well, or the types of students we have just don't handle very well, but for some reason with us it is data questions. And that's been the same since I've been at our school, with every year. So I'd like to have a go at this. I'd like you to draw out this table, and that will take you some time, I know, and get ready for some questions which are coming up on the next slide. So if you pause me, copy down this table, which will take a couple of minutes, and then we'll move on. Right, so the first question is to fill in a percentage. I anticipate nobody having an issue with that, but have a go at this, there is an issue people will have if you're not careful. So, fill in the percentage non-disjunction cells. The mistake people make is to do 1 divided by 1,000 times 100, which will give you a percentage, which will give you a percentage of 0.1%, is that? Yeah, fine. That's wrong, because if you think about it, there are actually 1,001 cells, 1,000 of them are normal, non-disjunction there is 1, and so it's 1 divided by 1,001 times 100. Similarly, this should be 3 over 853 times 100, 2 over 1,104 times 100. You should at no point be doing 3 over 850 times 100, because then you have imagined that this is all of the cells, when in actual fact this is just a subset. So the sum you should be doing, for instance, for 50 is 71 divided by 721 times 100. Well, the question then is, why did the researcher need to calculate the percentage? Why could he not just use the data? Why could he not just say that at 25 there was 1, at 30 there were 3, etc., etc., etc.? The reason is to make it comparable. 1 mark to make it comparable, to make it easier to compare. Second mark is for saying because there are different numbers of cells from each age of woman. Fine. So, for the next question, we need to understand this experiment. This guy has got egg cells from women at different ages, and he has counted how many of those eggs from a lot of different women are containing the wrong number of chromosomes, i.e. they have non-disjunction, and how many are normal. And then, of course, the reason for doing this is to work out, does the chance increase as you get up in age or go down in age? And so, the researcher concluded that there was no real evidence of a correlation between age and non-disjunction. Evaluate this conclusion. So, I've just got the answers on the board. So, those are your answers there. Hopefully that's quite nice and straightforward. Hopefully that's what you got already. Now, with the evaluate question, what we need to be doing is saying positives and negatives of this conclusion and what you overall think. So you should spend 3 or 4 minutes on this. It's actually a paragraph of work. It's at least a 3-mark question, if not a 4-mark question. Obviously I've made it up so it doesn't have a mark scheme, but in the real exam it will be at least 3 marks. Let's have a go at this, just pause me and have a go. Okay, so evaluate that conclusion. So positives and negatives. The positives of this, let's do negatives first. So the negatives of this conclusion are that initially, at least from 25 to 45, there's no clear correlation. It goes up and then down and then up and then down again. So that might lead you to think that there's no correlation between age and non-disjunction. The negative to this is, sorry, the positive to this statement is that at 50, there is a sudden jump in percentage of non-disjunction events by about 20 times. And that might lead you to think that there is a certain age at which you would increase your likelihood of getting non-disjunction events. You might want to say in your answer that in order to know, you would need to have more data on what happened at 55 and 60. And that would be perfectly reasonable to place into an evaluation. And in order to get a final mark for this evaluation, be it the 3rd or the 4th mark, you have to give your own conclusion. So you have to say, I do think it's caused by it increases with age because I can see it goes up dramatically at age 50, or I don't because it seems to jump all over the place. Both are perfectly reasonable. And as it turns out, there is a very strong link between the age, particularly of the mother, and the likelihood of this kind of non-disjunction event. So you'll find that for any condition which is caused by non-disjunction, be that Down syndrome, Turner syndrome, or any other, there is an increase in likelihood, particularly over the age of around 45. So let's do one more question, a little bit more wordy and less mathsy. That one. Now, I think this is a bit long. I'm looking at this thinking it's a bit long. So what I'd like you to do is to have a go just at B, C and D. So it's 9 marks. It's still quite long. In a real lesson I'll get you to do all of them, but I recognise meiosis was very long last time. I think we could do with a shorter lesson now. So let's just do B, C and D. It should take you 8 or 9 minutes. So have a go at those 3 questions. Just pause me and then that will be the end. Right, so let's have a look at the mark scheme. So describe what happens to chromosomes in meiosis. You need to write down everything you can think of. So, mark scheme. The chromosomes shorten, thicken, condense. That's prophase 1. The chromosomes associate in pairs. You should use the word homologous, but that's fine. And that's the second mark. Or the formation of bivalence, which is the same thing with a fancier word. Third mark, crossing over. Fourth mark, attaching to spindle fibres at the equator of the cell via the centromeres, moving to opposite poles, and then number 8 is effectively the whole of meiosis 2, that the chromatids are separated in the second division. Now there's 8 possible marks there, and for 4, 5 and 6, if you just use the word independent assortment, you can get a mark for that if you didn't get any of 4, 5 or 6. Fine. C and D are what are related to our lesson today, though of course this is very related to meiosis. A failure of the homologous chromosomes or chromatid arms to separate correctly, so actually you would get a mark for homologous pairs, leading to a loss or gain of a chromosome in the daughter cell. And why will this likely affect the organism? Because each chromosome has many genes present on it. Fine. That's the end of that lesson, and it's been quite a short one. Hopefully that's been quite straightforward for you. Really all you need to understand is this first slide, which we went through all that time ago, here. But you need to be able to apply it to data questions and to written questions. It doesn't come up enormously much, but it is a nice easy question when it does come up. I hope that's been useful for you. Any suggestions or improvements you'd like to make to these videos, I'm always happy to hear them, but given that I haven't heard from anybody, I am assuming that everybody is finding it okay. Right, looking forward to seeing you whenever I'm capable, and I will no doubt speak to you in the near future. Have a good afternoon.